In this presentation, we're covering Chapter 3, the kidney. Before you watch this presentation, I strongly encourage you to watch the following 3 videos. They're all very short, and they provide basic information which will help you understand the material in this presentation better. This chapter focuses on the anatomy of the renal system, and it goes into great detail. This presentation will cover the basic structures and operation of the renal system. I want you to focus on your learning objectives as you view the presentation. Also use your objectives to guide you to the appropriate sections of the textbook to read. The goal is to acquire knowledge of the overall system and how it works. The renal system has 2 functions: filtration of the blood and collection of urine. Filtration removes waste products from the blood, which, if they get to high levels, can be toxic to your body. It controls the body's fluid balance. We don't want to lose too much water. That will lead to dehydration, but we don't want to retain too much water because that leads to swelling. It maintains electrolytes: sodium, potassium, carbon dioxide, and chloride. If we lose too many electrolytes, this can also cause serious health problems in the body. All of the blood in your body passes through the kidneys several times a day. And the function of the kidneys, one more time, is to take in blood and filter it and put out urine. Understanding the process by which this occurs requires a basic understanding of the anatomy of the kidneys. Your body has 2 kidneys, and these are bean-shaped organs located on either side of your spine, below your ribs, and behind your belly. And each kidney is about the size of a large fist. There's 2 ureters, which carry the urine from the kidneys to the bladder. There's 1 bladder, which is a muscular elastic sac which collects the urine until expulsion, and 1 urethra, and this is the tube which transports the urine from the bladder to outside of the body. There are 2 anatomically distinct areas of the kidney, the cortex and the medulla. Glomeruli located in the outer cortex, and this is the exclusive site of plasma filtration. This is where the blood is actually filtered. The inner layer, or medulla, consists of pyramid-shaped tissues. Each pyramid has a duct that enters a calyx. The calyces funnel urine from collecting tubules to the renal pelvis. Urine flows from each renal pelvis and enters a ureter, which is about 25 centimeters long. The urine then passes from the ureter to the bladder, which serves as the holding area. And when about 150 mL of urine accumulates, a nerve reflex signals an urge to urinate. This is called the micturition reflex. Contraction of the bladder and relaxation of the urinary sphincter push urine into the urethra, which is 4 centimeters long in women, 24 centimeters long in men, leading to the outside of the body. The definition of micturition is the discharge of urine from the bladder via the ureter. In layman's terms, we may use the terms pee. So this is an illustration of the anatomy of the kidneys, where they're found, and the structures that are involved in the urinary system. The single functional unit within the kidney is the nephron, and is situated in 2 parts of the kidney: the renal cortex outer layer and the renal medulla middle interior of the kidney. There are 1.3 million nephrons in a kidney, and there's 2 major components to each nephron: the glomerulus, this is the specialized region where filtration of the blood takes place, and renal tubules, which are uniquely designed to allow the body to keep those things it needs and eliminate the rest as urine. Each nephron has 5 distinct areas, and I want you to know what these are and what the function of each one is. The glomerulus, the Bowman's capsule, proximal convoluted tubule, distal convoluted tubule, and collecting tubule. You'll need to be able to diagram these and or label these on a diagram. The glomerulus is the specialized region within the nephron where filtration takes place. This consists of a capillary tuft surrounded by Bowman's capsule. Bowman's capsule is the beginning of the renal tubule, and it collects the plasma ultrafiltrate. The proximal convoluted tubule begins at the glomerulus and takes a circuitous route through the cortex. Eventually, it straightens and turns downwards, and it enters the medulla to become the loop of Henle. The loop of Henle is the descending and ascending portion of the loop. So the proximal convoluted tubule goes down, and right before it reaches the bottom, it becomes the loop of Henle. The loop of Henle goes down, comes across, and starts going back up. Where it starts going back up, that is the start of the distal convoluted tubule, which is the ascending portion of the loop. Reabsorption and secretion of small molecules take place in the tubules, and also the reabsorption of water to concentrate the urine. The distal convoluted tubules join to a shared collecting tubule or duct that conveys the urine produced in several nephrons through the medulla for a second time, so a second filtration of that fluid occurs. The collecting tubules fuse to become larger ducts, and they empty into the calyces of the renal pyramid, and finally they empty into the renal pelvis. So spend time with this diagram. It's in your textbook, and again, be able to identify those 5 structures we just talked about. I want you to spend time studying this diagram, be able to label the structures that we just spoke about. This again shows you how blood is filtered. It goes into the Bowman's capsule, then it goes through the proximal convoluted tubule. It descends to the loop of Henle, which again, this is the portion of the proximal convoluted tubule, then the loop at the bottom, and then the distal convoluted tubule ascends, and then it goes into collecting ducts, and then goes down into the renal pyramid. Again, as urine moves through the tubules, these tubules join together, they become larger and larger, and then they empty into the renal pelvis. The urine passes from the renal pelvis into the bladder through the ureters, and then excretion of the urine from the bladder goes through the urethra. The kidney requires rich blood supply to execute its function. Each kidney is supplied by a single renal artery originating from the aorta, which the aorta is the largest artery in the body, and it brings the blood in at a rate of about 1,200 mLs per minute. That's incredible. The kidney is the only organ in which an arteriole subdivides into a capillary bed, then becomes an arteriole again, and then subsequently divides a second time into a capillary bed. And if you recall, as the blood travels from an artery, it becomes an arteriole, then it becomes capillary, and then that blood then continues on into the venules, which then those become veins. And it's all about size, with the arterioles being the smallest portion of an artery. The afferent arterioles carry blood from the renal artery into the nephrons. The varying pressures within the renal circulation cause the formation of the ultrafiltrate. So because of the changes in pressure, some things are pushed out of the fluid and becomes the urine. Others cause it to go back and be retained by the blood. Water, glucose, electrolytes, and other molecules less than 700,000 Daltons are allowed to pass out of the capillaries. They're small enough to pass through. It's a semi-permeable membrane. The afferent arteriole carries the modified blood out of the glomerulus into the renal vein, and the renal vein takes the blood from the kidneys to the vena cava, which is the largest vein in the body. I like this illustration because it actually shows the blood flow. The arteries are red, the venous blood flow is blue, and you can see how that passes through the kidneys. Approximately 180,000 milliliters of plasma is filtered each day, producing a final urine volume of 600 to 1,800 milliliters. Urine formation consists of the plasma filtration at the glomeruli and then reabsorption and selective secretion by the renal tubules. Again, we don't want everything to leave the blood. It has to be selective. We're going to retain these important components, and then we want primarily the waste products and water to leave the body. The kidneys play a very important role in the removal of metabolic waste, the regulation of water and electrolytes, and maintenance of the body's acid-base equilibrium. There are three structures of glomerular filtration barriers: the capillary endothelium with its large open pores, the trilayer basement membrane, filtration diaphragms located between podocytes of Bowman space. The selectivity of these barriers is based on molecular size and charge, and it allows water and small molecules to pass rapidly through. The endothelium has a negative charge that large molecules have to overcome to pass through. The reabsorption and secretion mechanisms are exactly the same, and they differ only in the direction of movement. Reabsorption allows those things that we need to go back into the blood. Secretion allows those waste products that we do not want to be secreted into the urine. And the tubular transport mechanisms are either active or passive. The active transport moves against a gradient and requires energy. The passive transport is movement from a higher concentration to an area of lower concentration. Each solute in the blood has a specific transport system that may differ by the area of the tubule where it occurs. So the tubule selectively reabsorb substances necessary for body homeostasis and function. And homeostasis is maintaining the same, maintaining a healthy body. Water, amino acids, salts, and glucose need to be reabsorbed to maintain homeostasis. Does not reabsorb waste products such as creatinine. Secretion occurs to eliminate the waste and substances not normally present in the plasma, such as drugs, contrast media from an X-ray, and also to adjust acid-base status of the body. Most substances secreted, other than hydrogen, ammonia, and potassium, are either weak acids or bases from metabolic or exogenous resources. Metabolic are those things produced through your cells, they're living and breathing and metabolizing, or exogenous sources that are coming in from the outside. I want you to spend time with this diagram. It shows the filtration and reabsorption mechanisms, and it's labeled 1 through 4 so that you can see as the blood is going in, the filtration that's occurring, the elements that are going out of the blood, and the elements that are going from the blood into the urine. I want to re-emphasize passive versus active transport. Again, these are how molecules are either retained or expelled from the blood. Active transport is the movement of a substance across the membrane against a gradient, and the compartments separated by a membrane differ in chemical or electrical composition. And this requires the expenditure of energy either directly or indirectly to get the substances across the membrane, and it occurs across the cells. Passive transport requires no energy, and it's characterized by movement of a substance along a gradient where the substances will move from an area of higher concentration to one of lower concentration. Each solute in the blood has a specific transport system. This table is located in your textbook, and it tells you the location in the tubules where the reabsorption takes place. It tells you the method, whether it's an active or passive, and then the substances. I'm not going to have you memorize this, but I want you to have an idea of what is actually happening in the tubules. This table is also located in your textbook, and it's a summary of tubular secretions of important ultrafiltrate components. So it says the location and the substance that will be secreted. Tubular transport capacity is an important concept that I really want you to understand. And this has to do with the capacity for reabsorption and secretion, which varies depending upon the substance involved. The reabsorption continues until the maximal rate is achieved. So again, substances will flow out of the tubules, but then we want to recycle a lot of these, and so they will flow back into the blood. The renal threshold is when the plasma concentration of substance is so high that it can no longer be reabsorbed. The substance will then be detectable in the urine. And the one that we think about all the time is glucose. It has a high threshold. It appears in the urine when plasma concentrations exceed 160 to 180 milligrams per deciliter. And the disease in which this occurs is diabetes. At these levels, the ultrafiltrate glucose concentration is greater than the ability of the tubules to reabsorb, and so the glucose will be excreted and detected in the urine. Other threshold substances include amino acids, ascorbic acid, creatine, potassium, and sodium chloride. And again, this has to do, the renal threshold is where the concentration is so great that it will leave the tubules, but the tubules are unable to reabsorb all of it that has crossed. So that will then be present in the urine. This is a nice illustration of the filtration abilities of the nephron. So you can see as it goes through the proximal tubule, and then it goes down across the loop of Henle, then back up, different substances are being able to cross over the membrane and or cross back. Renin is a hormone which controls blood pressure and various other physiological functions. Renin is released in response to decreased arterial blood pressure or volume, decreased sodium or increased potassium, and vascular hemorrhage. Renin causes angiotensin formation and aldosterone secretion. Aldosterone causes kidneys to retain sodium and water. This will then cause the blood pressure to rise. Antidiuretic hormone, or ADH, is also known as vasopressin, and it controls water reabsorption in the collecting tubules. So water reabsorption in the collecting tubules requires the presence of ADH. This is a hormone that's produced in the hypothalamus, and it's secreted into the bloodstream by the posterior pituitary gland. ADH regulates absorption of water in the distal portion of the nephron, making the walls of the distal and collecting tubules permeable to water. As plasma ADH decreases, the water is retained in the lumen fluid, resulting in dilute urine. As plasma ADH increases, water reabsorption increases, and this will result in a concentrated urine. Antidiuretic hormone levels will affect different components of the urine. Insufficient ADH results in diabetes insipidus, an uncommon disorder causing imbalance of fluids in the body. And this causes passage of abnormally large volumes of dilute, odorless, or insipid urine. And this can result in dehydration. Excess ADH results in high plasma volume, low serum osmolality, and high urine osmolality. Osmolality has as a function of the concentration of the urine. So if it's low in the serum, that means a lot of elements are passing through and are present in the urine, which will cause increased osmolality in the urine. Low plasma sodium and higher than normal urine sodium. So the sodium will leave the circulatory system and enter the urine and be present there in high amounts. Regulation of blood pH is crucially important. The normal range pH is 7.35 to 7.45 in the human body. Very narrow range. If it gets too high or too low, this can cause significant damage to certain organ systems. 3 body systems are involved in the maintenance of the pH. The blood bicarbonate buffer system. These are buffers that are present in your body that balance the pH and prevent the pH from changing too much. The pulmonary system. Your lungs. You can exhale or retain carbon dioxide, which is an acid. And the renal system increases or decreases the excretion of hydrogen ions, which plays a crucial role in pH, formation of ammonia, NH3, and reabsorption of bicarbonate. Erythropoietin, or EPO, is a hormone produced in the kidneys, and this hormone stimulates the bone marrow to produce and release red blood cells when oxygen levels are low. So the body will detect when it's time to make new red cells, and of course this is usually an ongoing process, and this is the hormone that will again stimulate the bone marrow to produce those cells. Be sure to write down any questions you may have after you have reviewed the videos, reviewed this presentation, and read your textbook, and have your instructor answer those during the guided discussion.